Good afternoon to one and all. Today, on the occasion of this Longevity India conference, I, Debasmita Saha, will be presenting my work on an imaging method to study the stress granule formation mediated neurite disintegration during the late phase of chronic hypoxia. So, as we all know that the brain is comprised of millions of neurons and they are interconnected with each other. So, in the neurodegeneration case, the tau tangles formation will be happening in the neurites, which leads to the synaptic loss and cause the neurodegeneration. And these diseases are impacted millions of people worldwide, and they are mostly characterized by the calcium dysregulation, protein aggregates formation, and the cytoskeleton disruption. So, in the normal brain, under any chronic stress condition, leads to the formation of protein aggregates, which ultimately cause the neuronal degeneration and which leads to the neurodegenerative diseases such as Alzheimer, Parkinson, and ALS. So, as these neurodegenerative diseases are characterized by the pathological inclusion, so there will be the early pathological condition and the late pathological condition. In the early pathological condition, we have seen that there are some stress granule aggregates will be formed, but at the late phase, these will be there will be some aggregation of the tau protein, amyloid beta, along with the stress granule formation to make a pathological aggregates. So we are mostly focusing on understanding how these stress granules are formed in the neurodegeneration process. So this is one video of the iPSC cells neuron where with the blue light induction optogranules will be formed. And so what stress granule mainly is, so these are membraneless cytoplasmic aggregates which are formed under any stress condition to protect the mRNA, and they are mainly comprised of the TDP43, TIA1, G3BP1 and the translation initiation factors. And under chronic stress condition, these will form some toxic aggregates. So in any stress condition, SGs will be formed, but when it is under a prolonged stress, there are some disease-causing misfolded proteins such as tau, amyloid beta will be included in the SGs and will form the neuropathology. So my main problem statement is that to understand, because neurodegeneration we know that it occurs under the hypoxic stress condition. So my work is to understand that in a chronic hypoxic stress condition, how these stress granules are formed and how the calcium dysregulation is happening, and what is there any correlation between the calcium dysregulation and the SG formation. Also, whether the SG formation leads to some neurite disintegration as well as the disruption of the cytoskeleton. So, to understand all these phenomena, live cell imaging plays a major role. So, but the major challenge of this imaging that under a prolonged stress condition, imaging for a long time will be difficult, as it is important to maintain a humidified environment. Also, the morphological details to understand will be cannot be achieved. So, for this, we have studied it in a confocal imaging system, where we can see that some incubator has been set up and it has been integrated with a confocal microscopy to study these phenomena with the DIC optic image. With the DIC optics, we can understand how the neurite retraction is happening under hypoxia, and some neurite morphology can be studied. So based on this, my first hypothesis is that whether the calcium dysregulation and the SG formation together will lead to some large SG aggregates formation, and also whether it leads to the disruption of the cytoskeleton and which finally cause the neurodegenerative diseases. For this, we have applied the live cell imaging method in an in vitro neuronal cell culture and a chronic hypoxia model we are developing. So to understand how the neurite disintegration is happening and the SG formation along with the calcium dysregulation, and to understand the interplay between all these phenomena. So for this, my methodology was that at day 0, we are culturing the neuronal cell N2A cell line, and cells are seeded and they are cultured till day 3, and then these cells are co-transfected with G3BP1, which is a SG marker, and RCAM, it is a calcium sensor. Why we are using RCAM? Because when we are using fluorophore, it is a dye, so it cannot be studied for a longer time, so we are using one genetic sensor. And when these are co-transfected, then After 48 hours of incubation, these co-transfected cells were treated with the COCL2 because it can mimic a chemical hypoxic condition. And then with the live cell imaging, we are studying at after 12 hours of treatment, after another after 32 hours. We are considering 12 hours as early phase of hypoxia and 32 hours as a late phase of hypoxia, to study that how the calcium dysregulation is happening, SG formation, neurite morphology, and the cytoskeleton disruption. So first, these are some of my results. So first we tried to do it at a single cell level. So these are the control condition where no SGs can be observed. But at the COCL2 treatment, at the 12 hours, some small SG aggregates can be observed. But at the 32 hours, we can see some large SG tangles has been formed. Also, with the heat map images in the early phase, we can see that these small SGs are forming, but in the late phase, these SG aggregates are fusing together and becoming a large chunk. Also, this is one live cell imaging. So here we can see that earlier these SGs are small, but later stage they are aggregating together and becoming a large SG aggregates to be formed in neurites as well as in the cell soma. So to further understand whether it is there is any correlation is happening with calcium, we did one single cell calcium accumulation studies under chronic hypoxia. So at 32-hour time point, we want to see that how the calcium is having some dysregulation is happening. So in control condition, the calcium is being in a homeostasis level in the cell soma as well as in neurites. But under this hypoxic treatment, we can see how the calcium is getting accumulated in the cell soma as well as in the neurites. We can see the calcium is getting overloaded, and some disintegration can also be observed with the DIC images. So we can conclude that the calcium dysregulation can lead to neurite disintegration. Next, we want to actually understand like whether this accumulation is also having some effect on the formation of large SG aggregates. For this is a co-transfection study. One is a G3BP1 and RCAM. So under the COCL2 treatment, we can see that there is some co-localization is happening at a 12-hour time point as well as at a 32-hour time point. But to understand in a better format, we did the line profile analysis. So at 12 hours, we can observe that there is some co-localization is happening in the 12 hours of hypoxic treatment, but at 32 hours, there is strong co-localization can be observed with the line profile plots, as well as we did with the Pearson correlation coefficient studies. So at which says the similar thing that under the hypoxic treatment condition, there is a co-localization is happening at 12 hours and 32-hour time point. Next, we want to study it at a neurite level. So here at 12 hours, there is no co-localization is been happening in the neurite studies. But at the 32-hour time point, at the late phase, we can see some co-localization of calcium with stress granules in the neurites as well. Now we want to see that whether the SG formation is whether it is leading to some cytoskeleton disruption or not. For this, we have with the F-actin we are using for the cytoskeleton study. So at the COCL at the hypoxic treatment late phase, we can observe that where the stress granules are formed, the cytoskeleton disruption is happening. Next, we want to study at the neurite level as well. So in the control condition, in the cytoskeleton remains intact in the control condition, but at the COCL2 treatment, we can observe that it has been got disrupted in the cell soma as well as in the neurites. And here is also with the DIC images, we can understand that wherever the SGs have been formed, it has been formed near the bleb formation, and it will further leads to neurite disintegration. Now we want to see that because it is a aging base, so we want to see at the Ross accumulation studies and the chronic hypoxia at single cell level. So in the late phase, we can observe that with the heat map images that Ross is getting accumulated in the cell soma as well as in the neurites level. So we can also conclude that the Ross accumulation is also leading to neurite disintegration. Next, that because Ross is also has a role in the cellular senescence. So at the 12, so we are testing it that what is the cell state of the cells which are having SG, what is their cell state, that whether it is apoptotic or it is in the senescence study. So we are using propadium iodide for the cell death study. So at the 32-hour time point, we can see that when the cells are with the cells having SGs are not apoptotic, but the other cells are apoptotic in nature. So we can conclude that the cells which are having SG are in a senescent state in the chronic hypoxia. And finally want to see the neuronal morphology and the hypoxic condition. So in the early phase, the green arrow shows the intact neurites, but the red arrow shows some bleb formation in the neurites in the early phase. And then these are the DIC images of the population study at the 32-hour time points, where we can see that the cells are having intact neurite under control condition, but some disruption of the neurites is happening under COCL2 treatment. Further, we could study it with the, so this is a single cell imaging, where we can see that at the COCL2 treatment, how the neurites are getting disrupted under chronic hypoxia. So overall, so here is my conclusion. So finally, we could conclude that under control condition, the neurites are remain intact, and the calcium is in a homeostasis condition, and the cytoskeleton is also in intact level. But in the early phase of hypoxia, the calcium is small SGs has been formed, and in the early phase, and there is a minimal co-localization is happening between the calcium and stress granule. And then the neuronal blebs are also being formed, but the neurite length remain intact as well as the cytoskeleton. But in the late phase of hypoxia, calcium is getting accumulated, and also the large SGs will be observed. And there is a co-localization is happening between the calcium and SG. And the neurite, and there is a shortening of the neurite length, and the cytoskeleton also become disrupted. So under chronic hypoxia, we could finally conclude that it leads to the calcium accumulation, stress granule formation, cytoskeleton disruption, and overall it leads to neurite disintegration. So here are the contact details of my guide. And finally, I would like to acknowledge my advisor and my co-advisor, Dr. Lopamudra Giri and Dr. Saptashi Majumdar, and all my DC panel members and collaborators, and my bioimaging and data analysis lab. Thank you.
One question.
Was this an animal study or a human study?
This is in a cell line studies I am doing. Okay. Neuronal cell line.
Okay, so will this be taken forward?
Yeah, actually that's why we are trying to do it in an in vitro cell line to study actually the mechanistic pathway, that how the calcium and all these are dysregulating. Accordingly, we also study some drug efficacy. Then we want to do it in a ex vivo and in vivo.
Thank you.
Yeah. Okay. I think
Again, the cell. Is it human or
This is mouse cell line.
mouse cell?
Okay, so we had a pretty intense 1.5, 2 hours now. So I'd like to thank all the speakers once again for their time, effort.